Dear students, a very good evening to all of you. So today is the part 3 of INI-CET Biochemistry revision. So first of all, let me know whether I am audible. Are you able to hear me properly? I welcome every one of you to this session. Try to be interactive in the session. Guys, if you have not taken Plus subscription, this is the right time to do so. With Plus subscription, you get the advantage of daily live classes, and we have structured courses. If you are a NEET PG aspirant, we have structured NEET PG courses. If you are a FMGE aspirant, you will have FMGE structured course. Recently, we have introduced MBBS Prof 1, 2, 3, 4, everything. So first prof, second prof, third prof. So all the first MBBS subjects. So for that, we are taking courses. So with this, you get the advantage of live tests and quizzes, and you get unlimited access to the previous courses also. As you are all aware, PrepLadder is now a part of Unacademy. With iconic subscription, you get the advantage of PrepLadder also. These are the currently going promotional offers. Try to utilize these promotional offers for your benefit. And there is now 5-year, 4-year subscription. So this 4-year subscription is going to be particularly useful for MBBS students. And in the Unacademy app, there is a new feature called Raise a Hand. Using this feature, you can directly talk to me during the class. So if you have any doubt, earlier you used to just type your doubt. Now there is no need to type. You can directly talk to me during the class. Currently, this is available for users who are listening to the class in the personal computer. Very soon it will be available for all the users. These are all the batches which started today. So these are all exclusive clinical subject batches, and we have integrated dual educator sessions. So all these batches started today, and NEET PG high yield revision batch also started today. Target Next 2022 also started, and I will be taking biochemistry at the end of July. So July end, I'll be taking the class. So with this, we start today's class. Try to answer the first question. During ATP synthesis in mitochondria, protons move directly through which of the following? Tell me. Please give your answer. So Michel's chemiosmotic hypothesis that explains the mitochondrial ATP synthesis. This ATP synthase, that is complex 5, it acts by a mechanism called binding change mechanism. So you all know at least this much. So via binding change mechanism, ATP synthesis happens. Okay. So here through ATP synthase, how protons enter? Tell me. So Arulmozhi, excellent Arulmozhi. So the protons enter through this subunit, FO subunit. O in FO. You should not pronounce it as F0 because it is FO. O stands for oligomycin. Oligomycin is an inhibitor of electron transport chain. Okay, oligomycin is an inhibitor. So oligomycin inhibits this binding of H plus to the FO proton channel. F1 is the one which is involved in the ATP synthesis by binding change mechanism. Answer the next question. So during the electron transport chain process, electrons are transferred by which one of the following? Excellent. Excellent, Arulmozhi. Correct answer. So there are two mobile electron carriers in electron transport chain. They are ubiquinone and cytochrome C. Among these, ubiquinone is lipid soluble. It is a isoprene derivative, whereas cytochrome C is a hemoprotein. It is a heme containing small protein. Next question. Which is true regarding the final electron acceptor in the ETC? Tell me. Kulmit, good evening. So, Kulmit, we are taking biochemistry in the Unacademy app. Okay. So, last month, and this month also, I have already taken 3 hours of biochemistry for INI-CET. So, please have a look at that. If you need extensive revision, I will suggest you to take the plus subscription. Okay, Kulmit? Ganesh Ilangovan. Okay. Arulmozhi is saying D. Okay, that I think it is for the previous question, Arulmozhi. For this question, the correct answer is, as Ganesh is telling, it is option C. In complex 4, that is cytochrome C oxidase, right? It accepts the electron from cytochrome C and it reduces oxygen atom. Ultimately, oxygen is reduced to water. Okay, so oxygen accepts protons and electrons, so it becomes water. Clear? Excellent, Ganesh. Correct answer. What is the chemical nature of intrinsic factor of Castle? So, what is this intrinsic factor of Castle? It is involved in the B12 absorption. Free vitamin B12 cannot be absorbed from the ileum. B12 is absorbed in the ileum. So, in the ileum, We have receptor for intrinsic factor bound B12. Okay. So as Ganesh is saying, the answer is correct. So it's a glycoprotein. So intrinsic factor of Castle is a glycoprotein. Tamogna, you are also correct. It's a glycoprotein. What is the significance of this? So, there is pernicious anemia. So in pernicious anemia, what do you have? You have antibodies again, intrinsic factor. Okay, because it's a glycoprotein, right? The antibodies can be formed effectively against glycoproteins. Clear? Okay. Name the inhibitor of thymidylate synthase. Thymidylate synthase enzyme is involved in the synthesis of DTMP. So UMP is methylated to TMP. The difference between uracil and thymine is methyl group at the fifth carbon. So that is done by thymidylate synthase. So this particular enzyme is inhibited by which of the following drug? Ganesh, can you revise your answer? So methotrexate is an inhibitor of which enzyme? DHFR, dihydrofolate reductase. See, this is UMP, right? UMP. It is uracil. Okay. So 5-fluorouracil is similar to UMP. So Tamogna, correct answer. So 5-fluorouracil is a suicidal inhibitor. Okay. So it is a suicidal inhibitor of thymidylate synthase. So the answer is 5-fluorouracil. What about allopurinol? Allopurinol inhibits xanthine oxidase. It is also a suicidal inhibitor. Hydroxyurea, this inhibits, tell me, what is the enzyme inhibited by hydroxyurea? Can anyone tell me the correct answer? Hydroxyurea, it inhibits ribonucleotide reductase. So what is this enzyme ribonucleotide reductase? This is involved in conversion of nucleotide diphosphates to deoxynucleotides diphosphate. For example, ADP is converted into DADP, deoxy. That is done by this enzyme ribonucleotide reductase. So that is inhibited by hydroxyurea. Okay, this is one interesting question. Give me the answer. Which enzyme is not present in cholesterol biosynthesis? Which of the following enzyme is not involved in cholesterol biosynthesis? Asif is saying A, that is thiolase. Others are saying it is option D. Akshay is saying D. Ganesh, Tamogna. Okay. See, thiolase is involved in cholesterol synthesis. How? Acetyl CoA plus acetyl CoA. It becomes acetyl CoA. Then one more acetyl CoA comes and binds. Here you get HMG CoA. So this is thiolase. This is your HMG CoA synthase. Then HMG CoA is reduced to mevalonate. So this is HMG CoA reductase. Where do you find HMG CoA lyase? You find HMG CoA lyase in ketone body synthesis, not in cholesterol biosynthesis. Is this clear? Asif, your doubt is solved, I guess. So this thiolase is involved in, let me tell you, it's involved in cholesterol, it's involved in ketone body synthesis also. Okay, so it's involved in many pathways. So this thiolase, even it is involved in fatty acid oxidation also. So you will find thiolase at multiple places. The thiolase reaction is a reversible reaction. Good, guys. So currently there is a promotional offer. With 3 month subscription you get 1 month free, and with 12 month subscription you get 2 months free. Okay, so hereafter we will be discussing some questions on metabolism. Try to answer. So it's a question which requires your thought process. Fructose, it enters the glycolytic pathway, and where does it enter? Tamogna, Ganesh. You are all saying it is option D. Let us see. Arulmani is saying it is B. Akshay is saying C. Okay. Fructose is acted upon by fructose 1-phosphate. Then it is acted upon by aldolase B. Then it produces glyceraldehyde, 3-phosphate, and dihydroxyacetone phosphate. So fructose enters glycolysis either in the form of glyceraldehyde. Okay, wait. So here fructose 1-phosphate, na? So it is glyceraldehyde, which will be further converted into glyceraldehyde 3-phosphate. Okay. So there is one more step. Anyway, so it enters either in the form of glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. So none of the answer is Correct. So sometimes these kind of questions can come. So where none of the answer is correct, in that case, you should at least attempt this question. So whenever they found that okay, the question is wrong, the answers are wrong, so they will give grace mark. They will give full mark to whoever who attempted the question. Okay. So it's very important when you find that the question is somewhat wrong, you should attempt. So here the option should have been either glyceraldehyde 3 phosphate or dihydroxyacetone phosphate. Clear everyone? Got convinced? Okay. In this question, there is answer is there. Okay. So this question, there is a correct answer there. Try to answer this question. I don't know how many of you are aware of the metabolism of mannose. If you can answer, try. Otherwise, I will give you the answer. Okay. Ganesh, Tamogna, Akshay, Siddharth, Piyush, Singhania. Great work. Look at this. Mannose is first phosphorylated to mannose 6 phosphate by the enzyme hexokinase only, and it is just isomerized by phosphomannose isomerase to fructose 6 phosphate. So the answer is option D. Okay. Sohaib, Tamogna, Akshay, revise your answer. So the answer is fructose 6 phosphate. So mannose enters glycolysis in the form of fructose 6 phosphate. Okay. Okay. Another interesting question. I'll move so that you can see the questions. Try to answer. Okay, so Arulmoli is thinking it is D. Piyush is also thinking the same. Let us see what others are giving. Ramogna is saying it is option B. Okay, let me point out this. So, which cells the student is growing? Neuronal cell. Now, do you want to revise your answer or are you sticking with the same answer? Everyone? Do you think the answer will be different now? So, Siddharth is saying the answer is A. You know, starch is broken down by amylase, which is a glucosidase. Siddharth, Jagan, Akshay, you are all giving the correct answer now. Good. So you have to add some form of glucosidase, because in human body starch is broken down by amylase to maltose, then maltose is converted into glucose by the brush border maltase. Clear? So this is a neuronal cell culture. Neurons do not have amylase enzyme, so we have to provide glucosidase. Clear, guys? Hmm? So this is the correct answer. Okay, Ganesh, in this question, so you are asking, sir, what about oxygen for neuron? See, Ganesh, so this is a neuronal cell culture. Okay, so this is a cell culture here. So here we are not mimicking the exact brain condition, right? So this is a cell culture. You can maintain cell culture in anaerobic conditions. Okay, so there is even something called hypoxic chamber, hypoxia chamber. So maybe the student researcher is trying to mimic the hypoxia of the brain. Okay, so the only macromolecule that produces energy in hypoxic condition, that is anaerobic condition, is glucose. So you have to convert starch to glucose. Clear? Hmm? See, the prerequisite for this is anaerobic condition only now. Hmm? And glucose can produce ATP in anaerobic condition. Clear? So if you just provide aerobic condition, starch cannot be converted into glucose without glucosidase. Is this clear, Ganesh? Right. Answer the next question. Which is false with regard to LDL receptor? LDL is a lipoprotein which is abundant in cholesterol. It is involved in transport of cholesterol from liver, from liver to peripheral tissues. There is a hepatic lipase that converts IDL to LDL. So the LDL is rich in cholesterol. The peripheral tissues, they take up LDL through LDL receptor. Okay. Okay. So now let's see each and every option. Hmm? Its cargo is low density lipoprotein. So it's correct, right? LDL receptor, it binds to cargo, otherwise we can use the term ligand. So its ligand is LDL. LDL receptor obviously binds to LDL. It is shuttled back to the cell surface after it delivers its cargo. It's correct. Okay, so LDL receptor is recycled. Okay, so in this context, you might have known a protein called PCSK9. Do you remember? So we have drugs which are PCSK9 inhibitors. So PCSK9 is related to this LDL receptor recycling. Have you heard of PCSK9? Arulmoni, have you heard of this? PCSK9 is very, very important. You should be aware of this. PCSK9 inhibitors are used. Good. Excellent. Bishwadeep. Okay. So it is a protease. Okay. P stands for protease. So PCSK9, it is a protease. Okay. So just remember this much at least. Excuse me. So PCSK9 is a protease. Yes. Evolocumab. So Akshay is saying it is evolocumab. Okay. Evolocumab is the, yes. So the full name, as Arulmoli is saying, it is a proprotein convertase subtilisin and kexin type 9. Okay. But anyway, no need to remember the full. At least you just know that it is a protease. Okay. Good. Siddharth. Good, good. Nice. Once bound to cargo, it enters the cell via clathrin. So it enters via clathrin because the LDL receptor itself is located in cell surface pits known as clathrin coated pits. So that membrane area which is coated by clathrin, you might have known this already, clathrin coated pits. So clathrin is the one which is involved. Then what is this COP1 and COP2? So these are involved in vesicular transport. From the Golgi apparatus, we have to and fro movement, right? So in this process, COP1 to COP2. So for example, from ER to Golgi you have transport, right? Golgi to outside you have transport, right? So in this transport, COP1, COP2, they are involved. Okay, so these are all proteins only. Clear? So these are all coat proteins. So these are all coat proteins. COP, coat proteins. Clear? So they coat the vesicles. Okay, so no need to go too much into this detail. Okay, as you are medical students, I think we can stick to this much. Option D, LDL receptor releases LDL in the low pH. That is correct only, right? So endosome, it fuses with lysosome. So endolysosome, the endolysosomes, the pH is low. So this low pH, it enhances the release of LDL, cholesterol. And finally, the receptor is recycled. Okay. So hereafter, I think the question should be easy, which is false concerning facilitated diffusion. So this must be an easy question for all of you. So false. Excellent. Facilitated diffusion, let me give one example. Glucose transporters. These are examples for which glucose transporter? GLUT. So among the glucose transporters, GLUT is example for facilitated diffusion. So it happens through the concentration gradient. Okay. So it is used to move charged or polar substances. It requires the use of proteins in the membrane. Correct. Because glut. See, glut is a protein. It's a membrane spanning protein. Glut proteins, they span membrane multiple times. Hmm? Glucose transporters, they do not require energy. Okay. Whereas you have whereas you have SGLT. This SGLT is involved in secondary active transport. Okay. So it indirectly utilizes the energy spent by sodium potassium ATPase pump. Clear? So SGLT is as example for secondary active transport. Ganesh, it is a secondary active transport. It's not primary active, it's a secondary active transport. Right? Answer this. So we have already learned what is oligomycin. We have learned that it's going to inhibit the FO subunit of complex 5 ATP synthase. So if someone takes oligomycin, what will happen? Vishwadeep. Excellent, Vishwadeep. So it's correct only, Vishwadeep. The answer is 1 and 3. Oligomycin is not going to inhibit the proton pump. Okay, it's no way related to the proton pump. It's just going to inhibit the complex 5. So ATP production will be decreased and there will be a higher concentration of protons in the intermembrane space. Okay, proton pump will be functional. It's not completely dysfunctional. Once the protons are that much accumulating, right? So if the protons are accumulating, then it may get slower. Hmm? So the answer is C, correct. Next question, which of the following can freely diffuse through the cell membrane? So here is the answer. Okay, so here is the option. See these options and answer. Why there is so much confusion in this? See, K plus cannot freely diffuse, na? So it comes through only channels. H plus, it cannot freely diffuse. Anything which is charged cannot freely diffuse through the membrane. Glucose, it's a big molecule, polar molecule, right? So polar molecule cannot diffuse freely. So oxygen, which is non-polar and it is a small, so a small gas can freely diffuse. So the answer is option A. Okay. Hmm? Right. Alcoholics often present with a deficiency of vitamin B1. What can you conclude about an alcoholic with thiamine deficiency? What do you think is the answer? So thymine deficiency, pyruvate is not converted into acetyl CoA. So he or she will not produce acetyl CoA from the breakdown of carbohydrates. So this is also somewhat correct. Pyruvate which accumulate will be converted into lactic acid. So in thymine deficiency, there will be lactic acidosis. So option 2 is for sure this is correct. Okay, option 3 is kind of partially correct, but option 2 is sure shot correct. So wherever it is option 2, that should be correct. So in only one place it is option 2 is there. See how I derived the answer. Okay, you may have some doubt about option 3 because they are saying that will not produce acetyl from the breakdown of carbohydrates. It's not going to be like that straightforward, but still. So option 2 is correct. So there will be buildup of lactic acid. So this is correct. Clear? Pyruvate can be produced. Pyruvate is how it is produced. It is produced by the reaction catalyzed by pyruvate kinase. Pyruvate kinase reaction does not require thymine pyrophosphate. So the answer is option B. Okay, answer this one. Which sequence is most likely to be a recognition site for a restriction enzyme? You just have to identify a palindromic site. Just select a palindromic site. Excellent, Abhishek Tripathi. The answer is C. So you see G, C, A, A, sorry, A, T, the complementary sequence, A, T, complementary sequence. So see, now see, this is G, C, A, T. Now it will become A, T. Put another A and T. Now again A and T. Is this clear how you write the palindromic sequence easily? So it's very simple to write the palindromic sequence. Any sequence, you can just write palindromic sequence. See, T, A, right? Reverse T, A, A, T, A, T, T, A, A, T. Is this clear? So you can write palindromic sequence simply. GATC sequence, G, A, T, T, C, GATC, right? G, C, A, T, A, T. So this is the sequence for eco R1. Okay, so this is the sequence. Right. So guys, I hope you found this class beneficial. So we have discussed some of the basic concepts, some tricky questions also. So with this, I finish this month's YouTube sessions. So we had 3 sessions consecutively and many of you participated with a lot of enthusiasm. I encourage you to go for plus subscription. There you will get more benefits than these classes. Thank you all for joining. See you in some other class. Good night, everyone. Happy learning. Let's crack it.